Okay, so yeah, just like before.
Make sure that the people are on the Zoom, and also make sure you videotape it.
Right. Yeah. So you can see it. It's recording.
Yeah. Yeah, I can see the people.
Good.
Yeah, it looks good.
Should we get?
I can see it.
Okay, I don't need an introduction.
You are already well famous.
So today I wanted to discuss with you, up to now, I'm going to give you a brief review of the first two lectures, because in some basic sense, they will explain what is happening in the third lecture. The first lecture was on the origin and evolution of metabolism, and it ended up with the citric acid cycle. And also the second lecture dealt with the evolution of the genetic code as manifested in the ribosomal proteins and in the aminoacyl tRNA synthetases. This lecture is now going to bring together the metabolism and the evolution of the code, and now we want to get at the origin of the code, how it originated and evolved in its early stages. Now, so I'm going to give you a brief overview of what I spoke about in the first two lectures. So the first lecture I talked about was my great discovery of the evolution of the genetic code from the GC code to a GCA code to a GCAU code, and its relationship to a metabolic metric. And as you could see here, how that evolution came about by reading a GCAU code instead of a UCAG code that was first written by Crick. And this was the metabolic metric, that is, the amino acids that are coming off of the citric acid cycle are coming off with lysine, alanine, proline, and arginine, and then glutamic and number 2, and then number 3, valine, leucine, isoleucine, methionine, etc. The interesting thing is that the first one is structural, the second one is hydrophilic, and the third one is hydrophobic. And I also talked about in metabolism from the bottom up of how the metabolic system had evolved from CO2, etc. And it was going through basically 3 stages. And it is the third stage, which I want to talk to you about, which is the Thioester world. That is a world where the metabolism was fundamentally built around thioesters instead of ATP.
I hate to interrupt.
The reduction was built around FESs and FES2s and not NADPH.
There's a lot of feedback and echo here.
Metabolism was quite orthogonal to the metabolism that dealt with when ATP came into the metabolic metric, into the metabolic system. So the reason that that's important, forget about all the rest, is that the synthesis of di and tri tetrapeptides formed from their amino acid thioesters. These initial peptides and their combinations have been able to provide additional catalytic activities to those of the iron-rich clays, which are the system that is evolving this metabolism. Now, the reason that that's important, there is a fundamental problem that anyone who deals with the origin of the genetic code has to deal with. You need catalysts in order to start the system, peptide or protein catalysts. The metabolic, system that's evolving is not capable of doing that. So the system has to have some help from outside. And so you don't go into a chicken and egg problem. You are evolving a system that can synthesize proteins, but at the same time, they are not capable of synthesizing the proteins necessary for them to evolve. It's a chicken and egg problem that haunts anyone who studies the origin and evolution of the genetic code. So that is the problem that I will try to answer in the later part of the talk. So the evolution of the translational apparatus was in lecture 2. This is the translational apparatus. There's the large subunit of the ribosome, the small subunit of the ribosome. There's the messenger RNA. There are the 3 little tRNAs with their, one has in the A site has an amino acid. The other one has the peptide chain attached to it. The third one is exiting. What happens now is the peptide chain is transferred to the one that has the amino acid on it, so it grows by 1 amino acid, and the peptide site now moves, the peptide tRNA now moves to the exit site, and the one that has just had the peptide attached to it moves to the P site, and a new amino acid with another attached amino acid will come in. So that's 3 of the components that have to evolve in order to have a translational apparatus. So the ribosome has to evolve, the tRNAs have to evolve, and the messenger RNA has to evolve. Now, what puts the right amino acid onto the tRNA? And also you'll notice that 1/2 of the tRNA is involved in the large subunit, and the other half is involved in the small subunit, and that's the one that is reading the codons. That means it has an anticodon on its end. Okay, so what we want to do is to look at now, can we get at the evolution of the ribosome and the amino acyl tRNA synthetases. Now, fortunately, there is a group at Georgia Tech that have done magnificent work on the root of the tree, significance and evolution and origins of the ribosome. So one of the things that we can now monitor in the evolution of the system is the evolution of the ribosome. How did it begin? How did it evolve? And what does this mean for the rest of the things? How did the messenger come into being? How did the small tRNAs come into being? How are the amino acids that are necessary to put on the tRNA? And how does the tRNA read the anticodon, the codon? So there are 6 phases that they have in the evolution of the LSU and the small subunit, tRNA, and proteins. 1, RNAs formed from stem loops and mini helices down here. Then comes the evolution further. In phase 2, the PTC is formed and catalyzes condensation in the absence of coding. That means it's not reading the anticodon. 3, the subunits gain mass, and at the end of phase 3, the interface is acquired and the subunits associated, mediated by the expansion of tRNA from a mini helix to the modern L shape LSU and SU evolution is independent and uncorrelated in phase 1 and 3. So this is phase 1, this is phase 2, this is phase 3, and that's phase 4, when the small subunit comes together with the large subunit, bringing together the PTC site, which I showed you, but the coding site is now joined together with the PTC. And the tRNA is evolving. So this is a beautiful paper, and I really recommend anyone who wants to study the origin and evolution of the ribosome to read this paper. Now, associated with the origin and evolution of the ribosome are proteins that are found in the ribosome. The ribosome is not just RNA. I've just showed you RNA evolution. And now the ribosome also has proteins. And there's a beautiful paper. I suggest that people go into this fellow's work more and more. Has to do with the ribosomal proteins as documented of the transition from unstructured peptides to folded proteins. That means when you start studying the proteins associated with this RNA system that's evolving, they are made up originally with peptides, and that these peptides are put together and they start inducing folding, because folding in proteins is a very difficult problem. So it starts off with peptides, and you could now read close to the peptidyl, the PTC site, which I showed you before, which is the oldest part of the ribosome. Proteins are extended and largely devoid of secondary structure. Further, from the center, their secondary structure content increases and supersecondary topologies become common, although the proteins are still lack a hydrophobic core. This is following the GC and the GCA and the GCAU code. You are going from structural proteins to hydrophilic proteins to hydrophobic proteins. And the folds are changing drastically when you go from the extended ones to the ones that have what is going to turn out to be beta hairpins and beta barrels. At the ribosomal periphery, supersecondary structures coalesce around hydrophobic cores, forming folds that resemble those seen in the proteins of the cytosol. Collectively, ribosomal proteins thus offer a window into the time when proteins were acquiring the ability to fold.
So are we, in this point in the story, is the ribosomal proteins, are there additional proteins performing other functions, or we're just ribosomal?
Well, that's the interesting question. No, at least these things are just following the proteins that are involved in the folding of the RNAs. That means they're not, so far, they clearly were proteins being made in the Thioester world, which were made out of amino acids and were performing catalytic functions of all kinds in metabolism, etc. These are a set of proteins that are being made probably in the Thioester world, because this system is now not ready to make full-scale proteins. But that is why the Thioester world is playing such a big role now in the evolution of these, of the ribosome, etc. Okay, I'm now going to go into the second big problem, which has to do with how do you put the right amino acid onto a tRNA, and how did the tRNA and these amino acyl tRNA synthetases evolve, and did they evolve? Well, here's what the aminoacyl, I showed you the aminoacyl tRNA synthetases. There are three major catalytic domains. There is the catalytic domain where the amino acid is activated by ATP and then put on to a tRNA, especially the CCA part of the tRNA, which I'll show you. And now the question is, how does the synthetase recognize what amino acid to put on the tRNA? That is, you have to recognize the tRNA and the amino acid, put them on together, and the question is, where does that recognition between the tRNA and the synthetase take place? And there are three regions two regions that you really have to notice. One is the catalytic region. The reason I say that is that in the beginning, the catalytic module, which activates the amino acid by forming an aminoacyl AMP, and then transfers the amino acid to the ribose of the adenosine of the CCA and of the tRNA. Then therefore, it's got to recognize something in the tRNA, which at that point is a very primitive. In fact, it may only be the CCA plus the discriminator site. The second thing is it moved from the arm of the tRNA, from the GC code, into the anticodon module would recognize. So the real recognition takes place in the movement from the GC code to the GCA code, because now you're expanding the amount of the amino acids that you have to recognize, and they're being recognized by their anticodon system in the tRNA. So now the thing is moving from the arm into the anticodon module. Okay, is that clear? And then there's the editing module, which is people put on the wrong amino acid onto the CCA, and you've got to recognize that it's wrong and edit it. I'm not going to deal with that so much, although I'll make one or two statements at the end. Now, there are 20 amino acids that we already established in the code. And there is one aminoacyl tRNA synthetase for each amino acid. Okay, so there are 20 of them. Now, what's interesting is that these 20 aminoacyl tRNA synthetases break up into two groups because of the difference in the catalytic module. One is based on beta hairpins, and the other one is based on alpha beta hairpins. So there are two groups of aminoacyl tRNA synthetases. And they are seen here. One group, class 1, is dominated primarily by hydrophobic amino acids. Leucine, isoleucine, valine, and methionine are all hydrophobic. Arginine, we already know, is a rather interesting compound, but it doesn't really, it's certainly not hydrophobic. And cysteine is a late entry also. So class 1, to some extent, in class 1A, is somewhat the majority of the amino acids in that one, leucine, isoleucine, valine, and methionine, 4 of the 6 are hydrophobics. And when we go to 2A, they are all serine, threonine, proline, histidine are all hydrophilics. Glycine and alanine are either have no side chain or a very weak hydrophobic chain. And then there's 1B, which is glutamic, glutamine, and lysine. And then there's 2B, which is aspartate, asparagine, and lysine. and then come the hydrophobics, the real hydrophobics, phenylalanine in 2C, and tyrosine and tryptophan. So when you look at this, these people did, they then studied the amino acid biogenesis, which is the metabolic code from the citric acid cycle for 16 of the 20 amino acids, evolution of the genetic code, and the amino acyl tRNA synthetases. They did this analysis. And amino acyl tRNA ensured the fidelity of the translation of the genetic code. That's where the genetic code started. Covalently attached the appropriate amino acids to the corresponding nucleic adapter molecules, tRNA. The fundamental role of amino acylation reaction catalyzed by the amino acyl tRNA synthetases implies that representatives of the family are thought to be among the earliest proteins to appear, especially if you need the code. Our findings suggest the organization of amino acid biosynthetic pathways and clustering of the amino acyl tRNA synthesis into different classes are intimately related to one another. A plausible explanation for such a relationship is dictated by early link between the amino acyl tRNA synthetases and the amino acid biosynthetic proteins. That is, there's a coupling between metabolism and the code. The amino acyl catalytic cores are highly relevant to the ancient metabolic reactions, namely amino acids and cofactors. So these proteins are related to the metabolic factor. In particular, we show that class 2A, which I showed you, class 2A, are mostly associated with the primordial amino acids, while class 1A, ARSS, are usually related to amino acids evolved later. Reasoning from this, we propose a possible chronology of the evolution of this thing. And here's their chronology. And here they give you the number of steps, etc. Why these are basically coming from class 1, are basically have a lot more steps involved. And then there's class 2, which is dominating both the GC code and the GCA code. And the reason that it's not completely dominant is that the glutamic and glutamine are in, if you look at it, glutamic, glutamine, and lysine are all the only ones in the GCA code. This is the GC code, the GCA code, and the GCAU code. And you're going from structural to hydrophilic to hydrophobic. So both the ribosomal proteins in the ribosome have shown this GC, GCA, GCAU evolution. They've gone from structural proteins to hydrophilic proteins to hydrophobic proteins. And now I'm going to show you the origin of the genetic code. This is the so-called operational code. The operational code was discovered by Hugh and Schimmel in 1988. That the major determinant in the interaction of the tRNA for alanine with the alanyl tRNA synthetase was the GU pair, a single base pair in the acceptor helix of the acceptor arm of the tRNA. This result stimulated Christian Dedew, who's, by the way, a big aficionado of the Thioester work, to write a second genetic code, still largely undeciphered, is written into the structure of the amino acyl tRNA synthetases enzymes that couple each amino acid to appropriate transfer RNA. The code is probably non-degenerate, will be older and more deterministic than the classical code. Here's the tRNA. This is what he is talking about. And is there a pointer? No. Okay. If you look at the arm of the acceptor arm, and you'll see the following things. You have the CCA, which is 74, 75, and 76. You have the discriminator base, which is 73. And then 370 is where the GU code for alanine is found. That GU at 370 is how the synthetase accepts that tRNA for alanine. And so they, Schimmel and Hugh then set about looking for these things with very small tRNAs, not the full tRNA, just arms of the tRNA. And they were all acceptable. And then you could also put in things with small loops, and it would also do the same thing. The loops made no difference. So that was a nice piece of work that was done by Schimmel. But then, in this paper, 4 primordial modes of tRNA synthetase recognition determined by the GC operational code, Rodin and Ono, then by studying the arms of numerous tRNAs, came up with an expanded second code, now called the operational code, in contrast with anticodons and also codons, which are built of 4 bases, G, A, C, and U. They're double-stranded precursors in the 1, 2, 3 positions of the acceptors appear as triplets, almost invariably composed of GC and CG bases. Even in the majority of modern systems, including the deep archaea, specific amino acylation of the acceptor terminus operates within the GC variety of the first 3 base pairs in the tRNA. The operational code found in the acceptor arm pointed to an archaic GC code. The general thesis has been argued that, like all other evolving systems, the code began simply and evolved to the present complexity. The very fact that the anticodon-codon-like triplets in the acceptor helix turned out to be composed predominantly GC and CG base pairs strongly supports this hypothesis. And they give the reference to Hartman in 1975. So the GC code, which was predicted in 1975, was found in the arm of the tRNA in 1997. Now, let's go back to the evolution of the ribosome. So you start off here, and in phase 1, RNA forms stem loops and many helix that begin to accrete. In phase 2, the PTC is formed and catalyzed condensation in the absence of coding. The SSU may have a single-stranded RNA function, and they don't know why that is. In phase 3, the subunits gain mass. At the end of phase 3, the interface is acquired, and the subunits associated, mediated by the expansion of tRNA from a mini-helix to the modern L shape. That's not quite true. LSU and SSU evolution is independent. It's completely independent. And in phase 4, the evolution of the subunits is correlated. So we go back to the tRNA. Again, looking at the acceptor arm, the CCA, and then the stem loops here. This is the anticodon, and this is the D arm, and this is the T arm. The T arm here is an exact copy of the anticodon. This one is a modified arm because they form the elbow. Okay, so we're back at looking at the tRNA. And then Rodin and Ono then invented, and with good cause, the how the tRNA was first formed. It was formed from small polynucleotides. And what they said was the first treaty is proposed to have been a short 3 or 4 polynucleotide. For example, XCCA 5'. X stands for the discriminator base, which could be either A or U. Rodin and Ono proposed a detailed synthesis of the acceptor arm from these things and from the codons, GGC and GCC. And here's their idea of what the arm looked like. It had an ACCA. This would be the discriminator base. This is the CCA that is found there. And then essentially you have the anticodons here, and then this UGGU and ACCA, which completes the arm. That's their model, having made this arm from this. And it's a very interesting conjecture. Okay, I'm going to introduce you to an enzyme that is so interesting because it is the only enzyme and the only protein that essentially codes for polynucleotides. Usually it's the case where polynucleotides code for proteins. This is the case where the proteins code for the polynucleotide. The story begins when the DNA polymerase beta was found to belong to an ancient nucleotidal transferase superfamily. And they had crystallized the system, and they found that this DNA polymerase beta was very, very similar to something called the kanamycin nucleotidal transferase. Kanamycin seems to be an antibiotic created by various bacteria that kill other bacteria. And it's inactivated by this enzyme that puts ATP onto the kanamycin and nullifies it. So it turned out that when they crystallized this DNA polymerase beta, they found that it had the same structure as the kanamycin nucleotidal transferase. And here is the structure of the kanamycin synthase, and this is the structure of the DNA polymerase beta. Now, what these people, Holm and Sanders, did is they found a peptide here with this structure in both in the kanamycin and also in the DNA polymerase beta. And it turns out that I'll come back to this, that they opened up that with this structure and this thing, it opened up that they found a whole bunch of proteins that was picked up by this peptide. And the most interesting one they found was poly-A polymerase. Now, poly-A polymerase is an enzyme that is operating today by putting poly-A on messenger RNA and determines how stable that messenger is. It's an enzyme that still exists. The people at Yale immediately showed that this is related to the CCA enzyme. And the CCA enzyme is something I'm going to go into in a minute, but I'm going to show you that this system that was picked up with the polymerase DNA, the DNA polymerase beta, belongs to a vast family of nucleotide transferases. And here, this is a paper by Kuhnen and Aravind. Here we describe what seems to represent the minimal domain of the poly-B superfamily and demonstrate independent existence of a minimal nucleotidotransferase, MNT, in a wide range of archaea and bacteria. And here is the MNT, and you could see it. It's related to the CCA enzyme and a whole bunch of other things. And I'm going to now show you what Wiener claimed. Wiener claimed that there was an ancient enzyme that then branched off into a CCA-adding archaeal enzyme and a poly-A polymerase in eukaryotic cells. There was a class 2 enzymes, which is a CCA-adding enzyme that is fundamentally bacterial, and the poly-A polymerase. There's 2 poly-A polymerases that are. So I'm going to talk to you about the CCA enzyme. Here they are. There are 2 of them. One is archaeal and one is bacterial. And I'm going to emphasize mostly the bacterial. The one thing that they have in common is the catalytic site. The head here is the same in both. Everything else is different. By the way, this raises an interesting question, which has to do with why is the genetic code so universal? It's absolutely universal. Yet, in the bacteria and in the archaea, you find quite different proteins evolved in the code. So this is the CCA enzyme, and it has a very unique property. It is also closely related to the poly-A enzyme. And the poly-A enzyme is a polymerase. It just makes a lot of A's, or it can make a lot of G's, or it can make a lot of C's. It's a polymerase. And this is a comparison of the bacterial CCA enzyme as related to the poly-B polymerase. And here you see something that's really interesting. That is, this thing here is the neck of the CCA enzyme, and it's coding for the CCA. That is, the CCA is now being coded for by the amino acids in the neck on 2 helices. And here's the way it's coded. It's hydrogen bonding, very much like a GC would. And here is a arginine. This is a glutamate, and this is I believe a aspartate. Anyways, this is hydrogen bonding to the CPP, and this is the catalytic site. And it's allowing these things to come in a definite order. Now, this is sort of show you without the helices and everything else. Here is this is the arginine site. There is the glutamic site. This is the aspartate site. And this is now recognizing the ATP. And here is the arginine site. Here is the aspartate, and there's the glutamic. And you see the hydrogen bond here. This is essentially recognizing these things by means of hydrogen bonding with the nucleotide, with the purine and pyrimidine bases. Now, what Alan Wiener then did, he essentially mutated this system and got a system that would now do things like it would not only do A and C, it would do U and G. And it would also do poly-CA. Okay, CCA enzymes. Now, the CCA enzyme, you would not survive without a CCA enzyme. That is because your tRNAs that are being made do not have the CCA on them. They have to be put on them by the CCA enzyme. And if you don't have a CCA enzyme, you don't have a CCA, and your tRNAs are useless. So, guided by that structure, we introduced mutations to reverse the polarity of hydrogen bonds between the nucleobases and the protein templates. We were able to transform the CCA into a UG, adding an enzyme to incorporate UTP and GTP instead of CTP and ATP. We transformed the related CCA and A enzymes into UU and G enzymes. And then we're poly-A polymerase into a poly-G polymerase. And we transferred this CCA enzyme into a poly-CCA by mutations in this other helix, which I'm not going to go into. So the CCA enzyme was creating the AGGA and the UGGU used to create the arm and the GC code found in the arm. So the question is, where did this stuff come from? It came from the fact that the CCA enzyme was creating these tetramers and trimers. Now it's even greater. What you can do is exchange the regions between the bacterial poly-A polymerase and the CCA adding enzyme. And what happens is sequence alignment suggests that the catalytic cores of both enzymes carry identical composites involved in nucleotide recognition and incorporation. This seems to be the prerequisite for the observed reprogramming of the catalytic order of PAP to incorporate sequence of defined length composition instead of long stretches of A. And you can now replacement of the C terminus of the CCA enzyme by the corresponding part of the poly-A polymerase generates a poly-CCA polymerase. So you now can not only polymerase A and G, you can now polymerase triplets. So the CCA enzyme is a rather interesting enzyme. And it is something that is probably involved in the origin and evolution of the tRNA, because it seems to have started with a CCA and an administrator site, whatever site. Okay, now there's another interesting fact, that if you look at the secondary structure of the CCA enzyme, which is on top, and the secondary structure of the class 2 synthetases, they are extremely similar, not in sequence, but in structure. And where do these, and they're made out of these beta hairpins, plus this motif too, and there's another motif in the CCA. Where do these structures come from? They don't come from the protein synthesizing machinery that now exists. They are coming from the thioester world. And that's the story which I discussed in the first lecture, that the third phrase about the addition of nitrogen created the thioester world, iron-rich clays with inputs of those kinds. Fixation of nitrogen occurred here on the surface of the iron sulfide, not pyrite. The amino acids were synthesized from the reverse citric acid cycle, giving rise to the metabolic metric. The number of catalytic steps in the biosynthesis of 16 amino acids from that cycle, and the 4 amino acids, phenylalanine, tyrosine, tryptophan, and histidine, are not derived from the reverse citric acid cycle. They are coming from sugar phosphate. The synthesis of di, tri, and tetrapeptides had been formed from their amino acid thioesters. These initial peptides and their combinations have been able to provide additional catalytic activities to those of the iron-rich clays. That is, the thioester world was providing the peptides that gave you the beta hairpins that essentially allowed you to begin the genetic code. Now, I just want to make one final statement, which has to do with peptides and how they may or may not have been involved in the origin of life, and how it is now a experimental system that now has to be developed to test these hypotheses. And so this guy Gazit from Tel Aviv University had a very interesting statement a fair time ago. The molecular basis of the origin of life is one of the most fundamental questions in modern biology. Of course, while the RNA world offers a very sensible model for the involvement of the current biochemical networks, there's a lack of knowledge about the early steps that led to the formation of the first RNA. That's right. The issue is as it is practically impossible that complex molecules as functional RNA oligonucleotides have evolved spontaneously. It has recently been demonstrated that peptide molecules as simple as dipeptides can assemble into well-ordered tubular, fibular, and closed cage structures. Other studies have confirmed that the ability of dipeptides to act as catalysts and capability of other peptides as short as tripeptides to serve as templates for nucleotide binding and orientation. Unlike complex RNA molecule, the spontaneous formation of functional short peptides in the primordial Earth conditions is very likely. We suggest a novel mechanism for the origin of life that is based on the ability of short peptides to form encapsulated structures, catalyst chemical reaction, and serve as a highly ordered template for the assembly of nucleotides. This model may explain the early events that led to the formation of the current biochemical machinery that combines the elaborate and coordinated interaction between nucleic acids and proteins to allow the functioning of living systems. So this now becomes a what I call a system of experiments. And so many eons ago, I did this experiment with a great group at MIT. The graduate student, Steven Santoso, was just unbelievably good. And Shugong Zhang is a very close friend of mine and a brilliant membrane protein thing. So we did self-assembly of surfactant-like peptides with variable glycine tails to form nanotubes and nanovesicles. The self-assembly of surfactant peptides containing 4 to 10 glycines as the component of a hydrophobic tails and aspartic acid as the hydrophilic heads is described. The peptide monomers form nanotubes and nanovesicles in water at neutral pH. These nanostructures become more polydispersed as the length of the glycine tails increases. These unique structures may serve not only as scaffolds for constructing diverse nano devices, but also as enclosures to encapsulate rudimentary enzymes for studying prebiotic molecular evolution. And these were the things that we used. These are the different tails with the aspartates. And this is what they formed. They formed these beautiful vesicles. And so what one could say, and this has been widely done. Okay, so what I want to propose is that this is now becoming a experimental system. It's not just simply talking about what might have happened, but the stuff that happened in biology today is an indication of what experiments to do. And I just want to end up very quickly now with the last thing. So far, I've talked a lot about the large ribosome. There is the problem of the small ribosome. What was it doing in the early stages? And as you recall, they are completely different. They're evolving parallel and not simultaneous, not together. And so the idea was that if you now look at the work of Rodin and Ono, you can now synthesize stem loops, which have the ACD and the GC code here, and also in the loop. So the stem loops now were now being synthesized. And now the stem loops can be utilized, both to attach an amino acid and be read at the anticodon level. And so there was the evolution, I think, of an independent protein synthesizing or peptide synthesizing machinery in a small subunit. There's a Shine-Delgarno sequence on there, and it binds the large RNA. I think that the first proteins that were synthesized were based on poly-G and poly-C, and there were polyglycines and polyprolines. They were performing structural functions. And then as they became more, when we went from a GC code into a GCA code, that was when there was a great transformation, when the code moved from the arm into the anticodon. And the synthetases now changed into these systems that now the evolution of the anticodon module now takes place. And for the hydrophilic cases, it was all based on alpha beta hairpins and beta barrels. When you went into the hydrophobic case, it was based on helical bundles. So the evolution of the anticodon module also goes along with the GC code, GCA code, and GCAU code. And so there, I think, is where I want to end. Now I want to make a few ending statements. It is clear that the origin of life and the origin and evolution of the code are evolved structures. They're not self-assembled. They're not based on an RNA world. They're not based on a soup. They're based on an evolutionary scheme that began with metabolism, evolved into the genetic code. The other point is that in order for these systems to evolve over such length of time, one thing has to be absolutely certain. You must get yourself into geochemical cycles. And the geochemical cycles are based on CO2 systems that are fixed, and then under sulfur systems, and then nitrogen fixations, et cetera, et cetera. So I would claim that if people are doing pixels looking for what's happening on planets away from the Earth, if you could pick up these great cycles, and what's even more important, if you can't pick them up, like you can't pick them up on Mars, it means that when these cycles break down, that's when life breaks down. And that is a clear point that every time in Earth's history where life has been challenged, it's been a breakdown in one of the great cycles. And so when I worked on the stuff that killed the dinosaurs, it was quite clear that acid rain was absolutely destroying the carbon cycle and stuff like that. And we are now living in a case where we are able to destroy these cycles, and our life on this planet are at risk. So I just want to end on that happy note.
Okay, fantastic. Thank you so much for this pretty exciting your lunch lectures. Any quick question from the audience? Very good. I think we all understood. So let us thank the speaker again.
Thank you.
Make sure that you have the recording saved.
I mean, I just.
Yeah. Okay, let's go to get something to eat.
Okay, thank you, and thank you. And so I did get it done.
Yeah, very good. Thanks.
Was it make sense?
Yeah, a lot of them.